viewer, welcome to our Glory Globe TV as a breaking news. Yes, researchers at the Mark and Maddie Stevens Neuroimaging and Informatics Institute, that is called Stevens INI, at the Keck School of Medicine of USC, have identified a previously unknown pattern of organization in one of the brain's most important areas for learning and memory. The study published December 30 in Nature Communications reveals that the CA1, a region of a mouse hippocampus, a structure vital for memory formation, spatial navigation, and emotions, has four distinct layers of specialized cell types, and this discovery changes our understanding of how information is processed in the brain and could explain why certain cells are more vulnerable in diseases like Alzheimer's and epilepsy. Researchers have long suspected that different parts of the hippocampus CA1 region handle different aspects of learning and memory, but it was not clear how the underlying cells were arranged, said Michael S. Bayankovsky, PhD, senior author of the study and assistant professor of physiology and neuroscience and biomedical engineering too. Our study shows that CA1 neurons are organized into four thin continuous bands, and each representing a different neuron type defined by a unique molecular signature. So these layers are not fixed in place. Instead, they subtle shift and change in thickness along the length of the hippocampus. And this shifting pattern means that each part of CA1 contains its own mix of neuron types, which helps explain why different regions support different behaviors. So this may also clarify why certain C1 neurons are more vulnerable in conditions like Alzheimer's disease and epilepsy. If a disease targets one layer's cell types, the effects will vary and call the vary depending on where in C1 that layer is most prominent. To reveal this hidden organization, the scientists used an RNA labeling techniques called RNA scope together with high resolution microscopy, and these combinations allowed them to see individual molecules to gene activity inside a mouse C1 cells and to distinguish different neuron types based on their genetic signature. In a total of 58,065 C1 pyramidal cells, they identified more than 3 lakh 30,000 RNA molecules, which are the genetic messages that indicate when and where genes are active. By tracking these patterns across the tissue, the team produced a detailed map that outlines the boundaries between different nerve cell types throughout the C1 region of the hippocampus. The analysis showed that C1 is not a loose mixture of cells but is instead made up of four continuous layers of nerve cells, each defined by its own set of active genes viewed in three dimensions, and the layers from extended sets that vary slightly in thickness and structure as they run along the length of the hippocampus. So this clear banded arrangement helps reconcile earlier studies that suggested CO1 was organized more like a gradual blend of mosaic of cell types. Amazing, sir. In this exclusive report, Glory Globe TV, Delhi, Bharat.